Hi, my name is Lauren Robinson, and this is my presentation for my biomedical data science capstone project where I use neural networks to analyze macrophage data. So I know this is a pretty broad audience. I figured I'd give a little bit of background on macrophages and the immune system overall. Um, so I'm pretty sure everyone knows that the immune system is responsible for fighting against any potential pathogens or invaders, such as viruses or bacteria that enter our body. Um, In particular, we have the first sector of the immune system, which is the innate immune system. And this is where you have all of your first responders to anything that has entered the host. And one of the cells involved in the innate immune system is macrophage. And macrophages do a variety of things, but one of the big things that they do is release chemicals when they detect a pathogen that recruits more immune cells, which leads to more inflammation, which allows the bacteria or the virus or whatever to be taken care of. And so I mentioned the word inflammation, and I'm sure there's different connotations that people have heard about that word. But the reality is that inflammation can be good and it can be bad. So it's good when we need to fight off a pathogen like a virus or bacteria, because of course we want to kill it and get rid of it so that it's not infecting our cells and causing issues for us. However, too much inflammation can cause an issue, um, whether it's because inflammation goes on for too long or it's too extreme of an immune response. These can lead to cell damage, tissue damage, conditions such as sepsis, where you have that really strong immune response, autoimmunity, where the immune system starts attacking itself or starts attacking the host, and allergies, when the immune system recognizes something as foreign that is foreign but isn't harmful and should not be attacked. So moving on to the experiment where I got my data from, this experiment, they observed the impact of a receptor called TGR5 on macrophage activity. Um, they had four different groups with two different cell types. You had wild type macrophages, which were able to express TGR5, and you had knockout macrophages, which were deficient in TGR5. For each of these cell types, they also had separate experimental groups where you had one group that was treated with listeria and one group that was untreated. What they found is that wild type macrophages had a lower immune response to listeria than knockout macrophages did. And you would think that you would want a higher immune response because you want to get rid of listeria because this is bad bacteria that you don't want in your body. But like I said before, too much inflammation is also bad. And that's exactly what they found here, where the knockout macrophages that didn't have high expression of TGR5 had a higher risk of sepsis. And so the clinical relevance of this is that we now know that TGR5 does play an important role in immune regulation, even when you want an immune response, but you want it controlled. And this is important because this information could be used in the future for drug developments, when you're trying to lower immune responses for various conditions. So moving on to what I did, I created a feed-forward neural network that could analyze the gene expression data and use that data to determine whether the macrophages had been treated with listeria or if they had been untreated. I used some of the data to actually train the model and then the rest to test the model. And I found that it was a success overall. So as far as future directions go, um, this model could be useful when trying to identify macrophage activity and potential infection in patients. If we can use these gene expression markers as biomarkers of um, Listeria infection or other bacterial infection, this could potentially lead to quicker diagnosis, which means quicker treatment and a decreased risk of someone actually experiencing sepsis. There are definitely things that can be improved in this model. I think it would be cool to also be able to train the model to decipher between the wild type cells and the knockout cells. So that's pretty much it. Thank you so much, and I hope this was insightful.